And I'm also a member of the Food Forum and the Planning Committee for this meeting. We are in this portion of the of session 3, we're going to focus on sensory and oral health. Our first speaker for this topic is Dr. Nancy Rawson. She is Associate Director and Associate Member at the Monell Center. Dr. Rawson is responsible for managing the corporate relationships of the center, supporting strategy development and intellectual property management, and as an associate member, conducts research in taste and olfactory cell biology. And her talk is on age-associated changes in the chemical senses. Thank you.
Thank you. It's a real pleasure to be here. This has been a really exciting forum. So my talk today, I want to kind of put a little bit of context around the chemical senses and really kind of bring it to where we're playing a part in terms of nutrition and nutritional status really is in influencing what and how much and how often we eat. The reasons for why we choose what we eat really are going on in terms of food preference in the brain. And the brain is getting information from both the external sensory stimuli, sensory input, and internal information from metabolic input, such as the nutritional status of the individual and so forth. This information really is kind of filtered through our experience, our health, and our age. And so it's not, these are not absolutes. We think that we know what salt tastes like or what a rose smells like. But really, these experiences are very different for each one of us. And these differences relate to our experience, our genetic makeup, and these other factors. And what's also interesting is that the metabolic input influences our sensory experience. Earlier, it was Dr. Frucci mentioned about insulin in taste, right? So we know that our insulin status can influence the turnover of taste cells. So it's a two-way street here. Not only do our senses tell us what kinds of foods we should be eating, but our internal state also influences how we're experiencing the sensory characteristics of those foods. The other really important thing I want to say is that our food enjoyment, our food preference, is really a multisensory experience. We think about taste and smell primarily, but there's also, for taste, there's many, taste has things like sweet, salty, sour. For smell, we think of fruity, smoky, toasty, floral, things like that. But we also have this chemical sense, what we call chemesthesis for things like hot pepper, stinging, burning, tingling. And the texture of food plays a very important role as well. Crispy, juicy, drying, and so forth. But we also know that vision and hearing influence our experience of food and our preference for food. You can very easily fool even a wine expert by changing the color of the wine. And what you get when asked to describe the wine, if you've taken a white wine and added red wine coloring to it, is descriptors that are typically only used for red wine. Okay. You can also change the person's response to a compound such as valeric acid or a sweat sock odor. If you tell them that this is from smelly sweat socks, they tell you they don't like it. If you tell them it's from a very expensive cheese, they tell you that, oh, this is wonderful. Right? Exactly the same chemical. Right? Okay. And similarly, crunch, slurp. Years ago, I worked for a Campbell Soup Company. We worked with pickles. And it turns out that the crunch of the pickle makes a difference in the perception of the flavor of that pickle. But when we think about aroma, even though we use the word taste often, it's really primarily, when we think about flavor, it's really primarily aroma. There's thousands of different flavors. And without the sense of smell, you can't tell wintergreen from spearmint or mango from a peach. So your food really becomes very uninteresting. And the taste or the flavor of food relies both on what we call ortho-nasal olfaction, which comes through the front of the nose, and retro-nasal olfaction, which comes through the back of the throat and up into the back of the nose. And this is very important if you're thinking about aging and thinking about mastication and how we chew our food. The duration of time in the mouth influences how much of this food's aroma is actually going to get to where the receptors are. And the receptors are up here in the upper part of the nose along the cribriform plate here. And this is old data, but it's still, all of the studies really are remarkably consistent, showing that by the time we reach about age 60, odor sensitivity has decreased by about 2.5 to 3 orders of magnitude. And there are gender differences with females declining a little bit later than males, but they also tend to start out a little bit better, typically, particularly with respect to odor identification ability. I also want to point out, though, that there is a lot of individual variation. And this is some data that we had collected, and each dot is an individual person, ranging in age from around 20 to 80-something. And while you can see that there is a downward trend in terms of, now this is dilution step, so these people are detecting a lower concentration than these people. You can see that there's a lot of variability. And there are people, even up here at 84, whose sensitivity was really identical to many subjects around age 20. And these were all generally healthy subjects, although they may have been taking some medications. This is, again, some older data, but very consistent, showing that odor identification ability declines. And there are, again, females tend to be better than males, and they start out a little bit, they tend to be a little bit better through middle age as well. I don't know how much of this, I think it would be interesting to look at how much of this is attention and experience and how much is biology. One of the newer findings is that in olfaction, it isn't simply the case that we become less sensitive to all odors equally. This is a more recent study by Thomas Hummel's group showing that they looked at odors that were higher in molecular weight versus odors that were lower in molecular weight. And so the heavier odors tend to be deposited more anteriorly in the nose versus the lighter odors that are more volatile and go up farther into the nasal cavity. And what they found was that the sensitivity to the high molecular weight odors, shown here in the elderly, was more affected by than the sensitivity to the lower molecular weight odors. So this is a low molecular weight, this is high. This is a low molecular weight, this is high in the older population versus the younger population. And so one of the reasons for this may be that the olfactory epithelium in the more anterior regions is more exposed to whatever nasty things are going up your nose. An anatomical study suggests that anterior region is where there's more patchiness in the epithelium and more infiltration of respiratory epithelium. So the receptors may just not be there in the older population. One of the really nice recent studies looked at the NHANES data, which incorporated olfactory data from 1,281 participants over 40 years. In this survey, they used an 8-item forced choice odor identification task. And they also looked at self-reported smell alterations and history of health, different types of health problems. And what I wanted to point out here was in the self-reported data, this is across decades for any smell alteration. This middle decade here, 60 to 69, is where people were most likely to notice that their sense of smell had changed. Before that and after that, they really didn't notice that there was any change. But when they measured their performance, measured dysfunction, the biggest changes actually were a little bit before that, 50 to 59, and then again, 70 to 79. So people do report changes, but they're not really tracking what we see when we actually measure dysfunction. And in the oldest group, the 80-plus group, nearly 40% of the population showed significant dysfunction in olfactory sense identification. And when we looked at the types of, dysfunction, a hyposmia is just an impaired sense of smell, and anosmia or severe hyposmia. Anosmia is absolutely no sensitivity, no ability to do this task. And you can see a strong increase here with age, and very little incidence of anosmia or severe hyposmia in the 40 to 49-year-old group. So the question becomes why? And some of the factors that were identified in this analysis, older male subjects are more likely to be Mexican American, lower income to poverty ratio, we've seen that in other talks today, poorer general health, which you would expect. The ones that I was kind of interested in were that they were less likely to be regular exercises. So the people with olfactory dysfunction were less likely to be regular exercises. So yet another reason to get your exercise. They were more likely heavy drinkers. This isn't too surprising that they've had multiple sinus infections. And I found this one also interesting, that they'd had wisdom teeth or tonsils removed or had had ear tubes. So the issue with ear tubes, I'm just going to leave it there. I think it might be interesting to talk about later. But those were factors that were identified as significantly associated. If we want to think about the biological underpinnings of this, a few points I want to point out. The olfactory mucus protects and transports odors across the olfactory epithelium. And I'm kind of surprised that nobody yet has talked about hydration status, but hydration is really important to maintain the right composition of your olfactory mucus as well as your saliva. And if this composition is too dehydrated, the odors simply cannot get to the receptors. And there's also a lot of individual differences in the receptors that we express among each of us. Another really important point is that the olfactory epithelium can, in fact, regenerate. It's a very effective regenerating epithelium. And we do see, even in the very elderly and in older animal populations, that the tissue does continue to regenerate throughout life. That's maybe a little bit of good news, although it certainly is not as good as it is in a younger subject when it comes to responding to injury or traumatic effects. So, but I think that the olfactory cells, understanding how this process occurs, can really help us to develop perhaps therapeutic approaches or ways of preventing olfactory loss. And we looked at both during early development and this process of progression through the epithelium from basal cell to progenitor and so forth up to immature neuron. And there's ways of obtaining olfactory epithelium from human subjects, from living human subjects, and you can culture the cells and you can grow them in vitro. And you can also look at their functional characteristics. One of the recent papers that I was interested in, we did hear a little bit about telomere shortening, and there was a paper where they looked at telomere shortening and found that it did impair regeneration from injury, but it didn't seem to affect normal recovery and replacement of the cells under normal homeostatic conditions. So that's kind of interesting area for further exploration. Also, just as one example, we know that retinoic acid plays a very important role in early development. And we had done a study, and a few other people have looked at the role of retinoic acid in differentiation of the OSNs, or the olfactory sensory neurons, throughout life. And we found that if you cut the olfactory nerves in a mouse and you administer retinoic acid just acutely, transiently, you dramatically reduced the time it took for that olfactory nerve and olfactory epithelium to regenerate. So I think looking at early developmental processes and the factors involved there can help us to craft paths, perhaps, towards therapeutic approaches. The other point I want to make is that activation prolongs the lifespan of neurons. We talk about, in the brain, we talk about use it or lose it, in muscle and pretty much everything else, right? And that turns out to be the case with olfaction as well. Both if you're looking at olfactory neurons growing in a dish or in a mouse or in a human olfactory epithelium, the ones that are activated have a longer lifespan than the ones that are not activated. And so recently there's been a number of groups that are doing olfactory training. And right now this has been targeted to human subjects with anosmia that have lost their sense of smell from a head trauma or from chronic sinus infections. And this is still in early stages of study, but it does look like there are opportunities for therapeutic improvement. So the studies that we've done looking at olfactory neuron function, we take olfactory biopsies from the superior aspect of the middle turbinate in living subjects. In this particular study, we looked at over 600 cells from subjects up to 88 years old, and we looked at to see, does the function of these cells change, or is it any different? And what we found out was really quite unexpected. In blue, I'm showing the detection thresholds for two different odors for these subjects. And in these blue lines for these two different odors, you can see the shape looks like the shape of the data I showed you earlier. There is a decline in sensitivity from around age 50 to the older ages. But in purple, what I'm showing is the number of individual olfactory neurons that responded to an odor. And what we saw there was exactly the opposite of what we expected. There were actually more cells, a higher percentage of the cells that we tested responded to odor stimulation. So it wasn't that the cells can't function, right? It might be that there aren't quite as many in these patients, but they were functioning quite well. And so we looked at it a little more closely, and what we saw was that we tested these cells with two different types of odor mixtures. And in the younger subjects, most of the cells responded to only one of the odor mixtures. But in the population of cells from older subjects, we got quite a lot of cells that responded to both odor mixtures. These were very non-selective olfactory neurons. And olfactory neurons typically express a single type of odorant receptor. And so what we hypothesize is happening is that the cells that are more active, they're more likely to be activated, have broadly tuned odorant receptors. And over time, those are the ones that become predominant in the population. So the implication of this is that there's a loss of selectivity, and there's going to be, basically you're going to have a harder time discriminating among odors because you've got receptors that are responding to a lot of different compounds. You'll still smell something, but you won't know if it's this thing or that thing. Right. So to summarize, sensitivity to odors declines, but the change is gradual and not universal. There's a poor ability to identify and discriminate odors. There's faster adaptation, slower resensitization, and self-report isn't a very good indicator of measured function. So I think my time is close to up. So I'm going to just summarize here. Taste. Taste is a little bit simpler. There's really some changes in all qualities, and there's some differences among specific taste stimuli. Again, discrimination, identification, and suprathreshold intensity is reduced. And most people with taste loss also have olfactory loss. So we have multiple sensory deficits that really are going to impact our response to food. So I think I'll end there. I'll be happy to answer any questions.